All right, we're going to get started, everyone. Hello, and thank you all for taking the time to join our webinar, hosted by myself and Jeffrey Meek with Veltek Associates. Today, we're going to focus in on identifying which products to use when writing your hospital cleanroom disinfecting SOPs, and also how both DiNovo and Veltek can be a resource in staying up to date with current USP 797 and USP 800 changes. We decided to have this webinar because based on some of the conversations that we've had with a few of our partners, we just felt that this is good information to review, and hopefully this can answer some questions and at least provide a little bit better light on what products you should be looking at and what the details are for writing your SOPs. So for now, I'm going to go ahead and pass it over to Jeff.
Hi, Brooks. Yeah, thank you for that introduction and inviting myself to come on here, represent Veltek, and kind of go through some of these things that that you mentioned there. So as Brooks said, my name is Jeff Meek, and I am the manager that heads up Veltek's healthcare sales division here in North America. And I just do want to point out that I am employed by Veltek, so you will see some Veltek products on these slides, but I'm going to speak in the most general terms I can, just so these concepts can be applied broadly to these topics across the things that we talk about. Also, this information is geared a lot towards, that I put together here for this particular one, towards hospital pharmacy and the 503A compounding space. So if some of you have like a GMP background, you may notice that there are some topics that maybe don't dive as deep into like those best practices of GMP. But again, I'm focusing more on that 797. But I'm happy to talk more about going further with best practices as far as like GMP requirements offline, if that's something you want to discuss, and how these concepts apply to that. And lastly, I'm going to focus mainly on 797. I'm not going to touch on USP 800 in this particular presentation because that requires its own special attention in of itself. So I'll go ahead and jump into this. So the agenda for this is we're just going to go through some of the general terminology here. I'm just going to touch on how these terms kind of come to be with these products. I know there's kind of a lot of confusion sometimes, like what's a sanitizer, cleaner, one-step cleaner. We'll kind of touch on that a little bit. And then we'll go through these different factors when to consider when you're writing your standard operating procedures. These are obviously not the only things that you need to look at, but they are things that I feel like they're important, but there are other ones obviously that you can consider, but we can get further into those. If you would like, feel free to shoot over a question if there's something that's not covered that you want to talk more about. And then lastly, I'll touch very briefly on the validation of chemical agents inside of a clean room. There are some other requirements and things that maybe a GMP facility does to help validate their cleaners, and just knowing what's going on in the market in that regard kind of helps you as well select those things. Key takeaways, again, just being able to classify these things, be able to assign contact times based on the manufacturer label claim, the goals that you have in mind, and understanding the importance of those factors for when you're developing those standard operating procedures as far as these products go. And again, just understanding the validations that maybe some other facilities are doing or how that can benefit you. So we have some important definitions. So there's more of them than this. But these are the ones that are the ones that you'll fall into or you'll see more often when you're looking through these products to implement into your contamination control program inside of a clean room, a classified area. So the first one I have on here is a cleaning agent. So people ask, is this a cleaner? Does it have a surfactant? Is it something along those lines? As far as for the definitions I have on here, all from USP 1072. It's just a guidance document by USP for disinfectants inside of a clean room space, pharmaceutical manufacturing space. A cleaning agent in their mind is it just removes residues or other things from the surface that could harbor microorganisms or inactivate sanitizing agents. As far as EPA is concerned, though, in those label claims, EPA, if it's strictly only a cleaner, EPA does not have any registration requirements. So if something is marketed as only a cleaner and has no sanitizing or disinfecting label claims on there, EPA doesn't really do anything to classify that as a cleaner versus not a cleaner. So in theory, if you're cleaning something off of a surface that's water soluble, water can be a cleaner if that's its purpose. But if you want to go into a step further as far as EPA definitions, there's the one-step cleaners and disinfectants. Those are regulated by EPA because they have those biocidal label claims that they need to validate. The next one you'll see, and you're probably familiar with, is a sanitizing agent. So, this is, for the definition, it's an agent that's reducing the number of organisms on the surface, fungi, viruses, bacteria, not necessarily a sporicidal. That's important to note. But the EPA, as far as to get this claim and put it on a bottle label, the EPA requires the manufacturers to prove at least a 3-log reduction of microorganisms, or CFUs, colony forming units, through standardized testing that's submitted to them that they approve. And that's a 99.9% reduction. So 999 out of 1,000 CFUs are killed in that test, and you have to prove that with that data to the EPA that they require. A disinfectant. This is disinfectant is essentially a sanitizer, but kills many more organisms. So it's it removes and kills all forms of harmful microorganisms when applied to a surface. The main difference between a sanitizing agent and a disinfectant is the EPA labeling requirements require a 6-log reduction for that to be on the bottle label. So that is a 99.9999% reduction of CFUs to get that label claim. So, and that's the standardized test that's equal across all playing grounds, for all the manufacturers out into the marketplace, regardless of the market that they sell into. I have the sporicidal agent on here and sterilant. I put these next to each other because they kind of interact with each other to an extent. A sporicidal agent can get an EPA registration if it's just registered for a C. diff claim. So that is a C. diff sporicidal. It's very common, especially in the 503A and the healthcare environment. Step further, and that is a 6-log reduction of bacillus or C. sporogenes. And those two organisms are incredibly difficult to kill. And the thing to note is you won't see these organisms really on a lot of label claims that you look at, because they're extremely hard to kill. And I think the shortest contact time out there right now for a product to kill bacillus at that EPA registered 6-log reduction is 25 minutes. So those aren't necessarily the two organisms that you look at on a label claim to classify a sporicidal agent inside of a clean room. However, in some manufacturing GMP areas, they do have to test against those, and I'll touch on that slightly later on. So going through and knowing those different definitions and what those requirements are by EPA to get those label claims, what's the contact time? That's a question that, as a manufacturer, and I'm sure as distributors out there with Brooks, they get asked these questions all the time. What's the contact time for this agent? Well, there's the sanitizing contact time and there's the disinfecting contact time, but there's some things when you're writing your SOP to kind of look at when you're determining this. And one of those big things is your environmental monitoring data. So what are the types and numbers of organisms that you're normally dealing with? The environmental monitoring for somebody, say, down in South Florida during the summer might look completely different than somebody in the Northeast during the winter. So I know that in the 503A and hospital pharmacy side of things, there's not a lot of strict environmental monitoring procedures that's required, but I do think that that's valuable data. And even the data that you get, every 6 months that you do that typically, or have your third party come in and do that for you, that data is really important to know, and then you can use that to help select your disinfectants, select your rotation schedules, things like that. Another thing that this can be really helpful for is, like, where are you seeing this? Where are you we're capturing these organisms? Are they in the ISO 5? Are they in the ISO 7? Are they in the anterooms? Are they close to water sources? All these things can kind of change how you are rotating and applying your products in these things. And then this is the big one for what's the contact time? So what's the number of CFUs that if you do isolate something in your environmental monitoring, what's the CFUs that you're getting? So if it's typically it's a very low number. So if you're below 100 CFUs in a clean room environment, that's pretty typical. That sanitizing contact time might be enough. You're never really seeing more than that. And also, if you're not recovering anything, that says something about your program as well. What does the product label say? So is it a sanitizer and a disinfectant? If it's both a sanitizer and a disinfectant, you have two options there. You have the 99.9% efficacy, or you can go for the 99.9999% efficacy. And that's really up to you on which you feel is best in your environment. You know, at a minimum, you need to be at that 99.9, that sanitizing contact time. But if you're at that contact time and your environmental monitoring is coming back good or coming back favorable, that's a sufficient contact time. And it is something that is referenced in USP 1072. So again, that's the guidance document for selecting these products inside of you just need a 3-log reduction of vegetative bacteria and a 2-log reduction of spores. So that 3-log reduction just happens to be coincidentally the requirements for a sanitizing contact time. So there are no EPA registrations for a 2-log reduction, unfortunately, but typically in those environments, they're validating that. So contact time, at least the sanitizing contact time, rely on your environmental monitoring results. That is typically our recommendation for a clean room environment specifically. And this is just a visualization of do you have to get all the way to that disinfectant contact time for a lot of these? This is taken straight out of the new proposed 797. We have, you know, the action levels for air sampling and also the action levels for surface sampling. Surface sampling in ISO 5 and ISO 7 areas, I mean, even if you're greater than 5 CFUs, you know, you have some actions that you need to take. So, you know, is a 99.9% or 999 CFU kill adequate? Yes. Would a 1,000,000 CFU kill essentially be overkill? You know, in a clean room environment, that's pretty. The answer would, in my opinion, be yes. So as long as you're coming in between those two contact times, you're getting to that at least 3-log reduction of those organisms. So here we get into a little bit of frequency of application. So this is a little bit newer here on the new revision of 797. So the big thing to take away from this slide and this rotation schedule is they changed the categories of compounding from low, medium, and high risk to category 1, 2, and 3. And depending on which one you fall into in your facility, they dictate a little bit more on what you need to be doing as far as frequency of your applications. The big change that I've seen as far as disinfectant applications, sporicidal application, is category 3 is now a month or a weekly application, whereas the category 1 and 2 remain a monthly. This is a chart that we put together. I based it directly off of 797. But we put monthly for category 1 and 2, or based off of your environmental micro monitoring results. So if you're seeing something that maybe justifies doing more frequent sporicidal applications, by all means, take that data and apply that. And then we have some Veltek products on the side. You know, one thing I like to point out, Degon Spore is a 1-step cleaner disinfectant sporicide. We just like to suggest to take that time at that monthly rotation or weekly rotation to do a clean of maybe the floors, walls, things like that, to just get rid of any residues that may be there, just a kind of a best practice, but not required. And this chart here is for the PECs and pass-through areas. This here is any sites outside of the pass-throughs or PECs. Counters, work surfaces, floors. Again, most of it's staying daily for your cleaning, disinfecting, for your daily broad spectrum. Whereas, again, those category changes kind of affect how you are, how often you're applying a sporicidal agent. So category 3, again, they're saying weekly now, where that's used to be the old high risk category. And category 1 and 2 is still monthly. And again, I put in there just or based off of your environmental monitoring results. Another thing to kind of note on this slide is the incoming supplies. So incoming supplies, there is a reference in there about wiping down incoming material with a disinfectant or a sporicidal agent. You can use sporicidal agents. They say you must use the sporicidal contact time. What they're referencing in 797, when they say sporicidal is a C. diff contact time. So there are products out there that have that C. diff contact time that you can use. I do like to point out, though, that alcohol in and of itself, if you're using alcohol to wipe materials going into the clean room, it does not kill spores. So just be aware of that. If you think that you are killing spores with doing that wipe down of your material going in with alcohol, that's not the case. We do have a 6% hydrogen peroxide saturated wipe that we'll recommend maybe over an alcohol wipe down for incoming material, just as a risk mitigation step. A higher percentages hydrogen peroxides are shown to have some sporicidal characteristics. They might not be EPA registered as such, but at least you have that added layer of protection when you're passing something into the clean room. A fun fact is, if you ever order a spore from ATCC or one of those services that will ship you spores for lab testing per se, they actually send you the spores in an IPA solution. So it keeps them only spore to throw out there. Something else to consider is your surface compatibility of chemical agents. So first off, when this comes up, you should ask the manufacturer if they have documentation on their products' compatibility with different surfaces. If they're a manufacturer that deals with the clean room space, just because of how sensitive that space is, they'll most likely have a validation report and a study that they can provide you showing the compatibility of their chemicals on the surfaces, common surfaces that are used in the clean room. So start there, ask your distributor if they can get it from the manufacturer, reach out to the manufacturer directly. They should have something. So, but that being said, all sanitizers and disinfectants, they do leave behind a residue. So all of them have to have those active ingredients or the guts of the product to work, some more than others. So I just listed here kind of the most common active ingredients or active formulations of disinfectants and sanitizers or sporicides that you'll see on the labels. First is alcohols, 70% IPAs, ethanols. They're very low in residues, but you do have to be aware if you're using these on large surface areas of the VOC risk, the volatile organic compounds. If you start applying these on large surfaces like floors and walls, you have the potential risk of those to evaporate, get in the air, and have some VOC risks. Quaternary ammoniums, they are very low in residues, but they still may require some quarterly cleaning to remove the residues from floors, walls, things like that. Not so much in the hood, because you are continuously applying alcohol in hoods, so you just naturally are getting rid of residues by doing that. Hydrogen peroxides, again, these are low in residues. Some have more than others. Some of them have buffer agents in there, so they may have slight residues, but I just say low in residues as a blanket statement. Again, these are potential depending on the concentration you're using. They could have a VOC risk associated with them. Sodium hypochlorite, these are bleach, essentially. Not essentially, it is. But these are high in residues. They're very effective sporicidal agents and disinfectants, sanitizers, very effective, but you have to be more diligent with your cleaning steps, especially in hoods and high value equipment like stainless steel and other surfaces, so they can oxidize those surfaces. We have hydrogen peroxide and parasitic acid formulations. These residues really depend on what formulation that you're looking at, or not formulation, but the active ingredients in the formulation. So some of these products manufactured by one manufacturer versus another may have higher percentages than another one. So just be aware of that. If that's the case, more diligent cleaning required. And if they are at those higher percentages, it does have the potential to oxidize some surfaces at that higher concentration, because it is an oxidizing agent. Phenolics, I put this last on the list for on purpose. They're high in residues. They're great for detergent. You know, they're historically been very good as detergency and cleaning, disinfecting. However, as a manufacturer, we have firsthand knowledge. We don't suggest these really to use for any new customers. Maybe for legacy customers, we'll say okay, but these are actually banned in Europe. There's some of the active ingredients in there that they feel is not safe for the environment. They've been banned in Europe, so we feel that's coming in the next decade to the United States. So that's something to keep in mind when you're selecting these. Sterile versus non-sterile. So this has been kind of a topic for debate with the version of 797 that's on the books right now. It doesn't really call out to use specifically sterile disinfectants and sporicides in the ISO 5 areas. Well, the new proposed revision of 797 does state that disinfectants and sporicidal agents must be sterile in the PEC, the PEX, and areas outside of the primary engineering controls. They should be sterile. I italicize must and should. The interpretation, that the answer I got on this language from USB, one of their Q&As, must is a requirement that they're putting out there. If they use the language must, should is a strong recommendation, is what they said in the answer that they gave there. So what does that mean for you when you're selecting these products? A question asked you, what's the risk of using a non-sterile product in the primary engineering control, or taking a sterile product out of the area where it's supposed to be stored to cleaning a lower grade area, then it going back and cleaning a higher grade area? So it's up to you. I think best practice is to use sterile in all classified areas, use a sterile product in all areas. That just eliminates the risk of any cross-contamination, using the wrong product in the wrong area, and having to write some type of deviation or come up with an action plan to remedy the situation. And then, you know, is it worth having multiple products to order depending on if it's in the ISO 7 and 8 versus the ISO 5? If you can have, you know, one SKU to order for all areas, that is an ideal situation. So those are two things asked. You can theoretically, per the USB guidance, you know, have a non-sterile product. It still needs to have some GMP or certs that you can reference, contact times, things of that nature. But those are just things to consider. And one thing to reference, I did put a note here. FDA does say that sterile disinfectant sporicides should be used in all classified areas. So this is referenced in their insanitary conditions document. It's a guidance for industry document. It's a pretty good read, but it does call out using non-sterile disinfecting agents in an ISO area as being is resulting in a Form 483 being given to a customer. So 503 facilities are not exempt from insanitary conditions. So if FDA for some reason comes in your facility, I know that's not necessarily likely for a hospital, but other compounding facilities have had that happen, and they've been giving citations for this. So just something to keep in mind. The chemical stability once opened. So again, ask your manufacturer about the chemical stability. They really should have validation reports that they've done studies to have this information available to you. So to ask them first. Don't spin your wheels, try to find it somewhere. Usually it's something you have to ask for. Sometimes they'll write out, send it to you, you know, if you're involved in a sales process with them. But if you're not, ask for that. They should have that documentation. One thing to note though is chemical stability is a lot easier for us as a manufacturer to give you definitive data on than also in-use sterility. For us at Veltek, we do have a report that takes both into consideration. And most of our products are good for 30 days if it's a bottled product, some bottled alcohols up to 60. And then aerosol cans, they last up to 3 years or so. They're a closed system. So, but from a sterility standpoint, it's tough for us to give you definitive data because that's more of up to how you're handling the product at your facility, your storage requirements, where you're opening it, things like that. So if you follow pretty sound practices, you'll probably get the same relative data that the manufacturer got when they send over their validation reports. But again, we can't determine the sterility 100% because if you open it in a different area versus another one, it could affect the sterility of it. One thing I do want to touch on is the sterile water. We get a lot of questions about how long is sterile water good for once it's open. Again, it goes back to, it's tough for us to say. It's more about how you are handling that. We do have documentation for all of our other products that they're good for at least 30 days. If you're storing that in a area that is of sound quality and you have good practices, you can mimic those. So that's good data to base sterile water off of. However, we didn't really do any testing, and most companies don't do testing for sterile water because there's no active ingredients, so there's nothing to degrade over time. So there's no stability testing to do. And at that point, it's really just up to how you handle it. So there's not a lot of data for sterile water, but you can base your in-use expiry off of the other products that manufacturer makes. And I'm not going to touch on these sections too much, but I want to point out some SDS language and PPE requirements that are really, well, where to find these and important sections when you're dealing with this. So section 3.2 shows it's each product. So if you go into the SDS and look at this, it might not necessarily reflect all of the ingredients that are listed on the bottle label. They may not be an active ingredient on the bottle label, but if they're considered potentially listed as a hazardous or some type of call out for these particular chemicals, they have to be listed on the SDS. So you might see some additional chemicals on the SDS that you might not see on the actual bottle label. So it's important to look at that. And then section 8.3 is another really important section. This is the PPE section, individual protection measures section. So when you're looking at this, make sure first you're looking at the concentration that you're using. Some products have different concentrations, but this will tell you if you need eye protection, respiratory protection, hand protection. And most of the time, obviously, these SDSs are worst case scenarios, so they're going to be as conservative as possible. So these are those requirements that are on there, and that's in section 8.3. And then the last section I like to just throw in there, it's another one that's good to just kind of look at real quickly, is the other information section. So this is where you'll find the health hazard ratings and the fire hazard ratings. So the higher those numbers, the greater the PPE requirements potentially, the more flammability risk it may have. So that's important to kind of just look at quickly, because if you're cleaning a large surface, those may be more important to you from a flammability standpoint. You want to clean, a large room, cover the whole floor with an ethanol-based product? Probably not. So those are things that are important to look at. So this section here kind of shows you that real quickly with a pictorial. And then I said, this is the last topic, and then we can get into some questions, but I kind of touched on at the beginning, validating chemical agents inside your clean room. So obviously the GMP side of clean rooms and compounding drugs and manufacturing pharmaceuticals, they have different requirements, but if you may ask like, how are they able to use some of these at a 3-minute contact time where I have to use it as a 10? On the bottle label it says 10. How are they get away with that? This is kind of what they do. This is part of their validation process. So like some of the questions, like what happens if your facility wants to use a product as a sporicidal agent and it has sporicidal characteristics? It's a bleach or something along those lines. It's GMP produced, it has all this documentation, but it doesn't have a label claim as a sporicidal agent. You could potentially validate it to do that. We'll talk about it on the next slide a little bit, but and then what if you want to use a product that had a lower contact time than what's on the label? How do you do that? Maybe you have a disinfectant. This is rare, but what if you have a disinfectant that has a 10-minute disinfecting time, but no sanitizing time? So you're not quite sure where the cutoff is. You could do one of these tests to validate that contact time against some challenge organisms. And again, what if you want specific claims against Bacillus subtilis or Clostridium sporogenes at a realistic contact time? Again, nothing out there really less than 25 minutes from a label standpoint. If you want to call out these bugs in particular, this is a way to do that. So what you do is you do disinfectant efficacy testing. You're not required to do this at all in a hospital setting or a 503A setting. It's something that some of the 503B outsourcing facilities have been doing. California, when you register with them, they've been asking for this documentation. Also, FDA at pharmaceutical companies will ask for this documentation as well. You don't have to do it, but it's something you could do if you wanted to use different agents or really kind of broaden the spectrum of chemicals that you may be able to use in your clean room. So really the definition of it is a study that confirms that disinfectants used against specific organisms are effective at in a practical sense, so practical use. And really what that means is clean rooms are specific. They're a specific environment. They get a lot of airflow changes. Long contact times might not be needed, but again, they might be overkill. So you validate these as a practical standpoint. And again, 1072 just states you need that 2-log reduction for endospores and a 3-log reduction for vegetative bacteria. So that's what they kind of look for with these tests in a clean room environment. But it's something that you can kind of look at if you want to, validate something else than just rely on the label claims. It is something that you can look at. But I think that is what I have so far. That is what I had. I think I had about 30 minutes. I think I came in just short of that, but we wanted to leave some time for questions at the end. So Brooks, I don't know if you saw any come through.
Yeah, we actually had 4 questions that came through. I'll read them off to you here, Jeff. Okay. So the first one is, do I need to rotate different disinfectants?
Okay, yeah, so again, 1072 has a good paragraph on this, a nice little section on it. So you don't need to, when we talk about rotating disinfectants, we're not specifically saying rotating like a quat for phenolic. What they're referring to is using a disinfectant and then rotating a sporicide. So it used to be that they thought that there could be some resistance built up to certain chemicals by microorganisms. That's never been proven to be true, and it's probably very unlikely, and they do touch on that in 1072. What they're really referring to is rotating a broad spectrum general disinfectant and a sporicide. And I will say they typically recommend that because most of them are pretty hard to work with, honestly, and they're maybe caustic on surfaces. So you kind of want to limit your exposure to them. So you use that broad spectrum daily, and you rotate that spore in to kind of get rid of all the extra stuff that broad spectrum might not get to. If you have a sporicide that kills everything and you're comfortable using that daily, or you find a product that you can use daily, you don't necessarily have to rotate in a disinfectant, but those are hard to find. So usually best practice is rotating disinfectant with a sporicide.
Thank you, Jeff. The next one's, can I use a 3% sterile peroxide solution as a sporicidal agent?
So we have so like 3% is kind of the percentage as a antiseptic. That's kind of what you see commonly at the drugstore, things like that. We do have a sterile version of that, but honestly, that is probably the weakest disinfectant or sanitizer that we manufacture at Veltek. It's going to have disinfecting properties and be able to probably be used in a clean room environment in that standpoint if you validate it, but not as a sporicidal agent. To have sporicidal characteristics, you really have to get up closer to 6% hydrogen peroxide. And a 3 just isn't quite strong enough to get there.
Okay. And the next one, how are you justifying using a sanitizing contact time when USP 797 requires disinfection? State board inspectors and accreditation surveyors will expect that the disinfection contact times be followed.
Sure. Yeah. So again, we go back to what is disinfection versus sanitizing. So it goes back to those EPA definitions. If something is both a sanitizer and a disinfectant, you're going to get a 99.9% reduction at that sanitizing contact time. And at the disinfectant contact time, you'll get the 6 log reduction, 99.999. USB in their guidance document themselves, USP calls out that in the clean room, you only need a 3 log reduction for vegetative bacteria. So that's the sanitizing contact time. That's all that 503Bs are mandated to get to when they do efficacy testing. So a disinfectant contact time is overkill for a clean room environment. So when you're doing disinfectant contact time, you're just following the EPA claim for the general test for everything. So that's the same test as if you're cleaning a gas station bathroom versus if you're using that same product or chemistry in a clean room environment. So it's really just looking at the definition of what they are and how many organisms they're killing, and really looking at the enumeration of organisms in that facility. I understand that it calls out using a disinfectant, but I think that's more of just a product category and not necessarily looking at it in a practical application in the clean room environment. If you have data that shows it has a 99.9% efficacy against gram positive, gram negative organisms, that's a broad spectrum that you can do and use, especially if your environmental monitoring is coming back good. So.
The next one, it's in regards to slide 8, Jeff. It says here is category 3 does not equate to high risk. You could do high risk compounding, non-sterile to sterile, as category 1 or 2.
Yeah. I was just speaking in general terms. That's kind of what the interpretation was at the beginning of 1, 2, and 3 were kind of changed to high, low, medium. Obviously, there are slight differences there, but I was just calling out that if you are doing category 3, that there are different requirements than category 1 and 2, and those go into your sterility of garments, frequency of environmental monitoring as well. So it's just kind of a lot different as far as this product group and contamination control goes. They require a little bit more per how it is right now. We'll see how it changes with comments or anything. But
okay, awesome. Well, Jeff, thank you for your time today. Glad to be partnered with Veltek, and hopefully everybody on this call or webinar today was able to have some questions answered, or at least provide some sort of clarity. And I know USP 797, there's been quite a bit of revision changes over the year. Jeff and I would, if there's any additional questions that anyone might have, or if you just want to talk a little bit further, we also have a kind of a sample or a sample SOP that's got some logging options in there, so you can kind of keep track of your daily, weekly, and monthly cleaning. We do have those available, so if you'd like to see more of that, please reach out to myself or to Jeff or to your DiNovo sales representative, and we'll set up a call. That way we can further review this. Again, thank you for everybody's time. This will conclude our webinar.
Yep, thanks everybody.